So as you walk in through the main gate, towards your left you are going to see a small entrance which is the connecting zone between the junior college and the senior college. So you will have to pass through this very beautiful garden that you see over here, and as you reach that end you will be entering the junior college corridor. So here I am reaching the junior college corridor. This is the junior college main gate from which also you can walk in, and this is the entrance of the junior college building, and here on the first floor is the microbiology department. So you have to climb the stairs, and as you climb the stairs, on the very first floor we have the entrance of the department. So here is the Department of Microbiology. This is the entrance of the Department of Microbiology on the first floor. You will see that here is the board which indicates the unique serving pursuits of the department. As you enter the department, on the first right you will see there is a classroom which is a microbiology classroom which we call as the seminar hall. This is the location where all our SY, TY, MSc lectures take place. FI lecture takes place in the main building because of the huge strength that we have. It happens in a classroom called as F11, which is close to the chemistry department, but all our SY, TY and MSc lectures happen in this particular lecture hall that we have in the department itself. As you come out of the seminar hall, you will walk inwards, and on the next right you have the storeroom. So storeroom is where all our non-teaching staff normally sit, and we have all our chemicals, all our glasswares, which are all kept over here in these cupboards. You can see that there are many cupboards all labeled, and all our chemicals which are kept in stock, they are kept over here, and all glasswares are kept in the cupboards down. Then as you move ahead, you will see we have two labs. It's called Microlab 2 and Microlab 1. Okay, but before we get into the labs, this is the preparation zone. So here all the preparation takes place. You can see that there is a weighing balance over here. All chemicals are kept over here. All liquid chemicals are here, and all solid chemicals are labeled and put in the cupboard. You will also find all your tubes and glasswares in these drawers which have been kept over here. And you will see that all media preparation, all chemicals that we use to cultivate microorganisms, all that preparation happens over here. And you have to use everything sterilized in microbiology lab so that there are no other microorganisms coming in. So this is an instrument called as autoclave, which you will be learning in your theory. So this is used for sterilization purposes. So we have 3 of them in the department. And at the same time, you will see here at the top, we have distilled water apparatus, which produces distilled water, which is free from all ions and any other, agents, because the water, the tap water goes into this, it's, boiled, and the steam is further condensed so that any impurities are removed from it. And that distilled water is used for all the preparation in the microbiology lab. Now let's go into the first lab, which is called as the Microlab 1. So Microlab 1 is the undergraduate laboratory. So as you get into the undergraduate laboratory, you will see that this is the lab where for the next 3 years you will be working. So you can see how the lab looks like. At every working station you will find burners, because you are expected to work between the burners. So the burners are normally maintained in an orientation like this, and your hands are between the burners, and you will be working between these two flames. So this is the lab where you will be working, and let me tell you that as there are burners, you cannot keep the fans on during the practicals. And it's a completely enclosed lab because it's a microbiology lab, and you know that microorganisms are everywhere, they are in the air also, so we can't let microorganisms from the environment enter into this lab. And that's the reason why this lab is completely packed and enclosed, as you can see. It's a huge lab, as you can see over here, and this is going to be the zone wherein you will be performing all your experiments when you are in your undergraduate studies. Now, as we move out of this lab, there is a similar kind of lab outside, which is called as the Microlab 2. I'm sorry if there is a disturbance that you can hear because of heavy rains that's taking place right now. So this is the postgraduate lab, a very similar laboratory of same kind that you can see over here, where our MSc students, our master's students work. They have their own cupboards for their chemicals and other requirements, and there are some additional instruments over here which are required for the master's studies. So this is the master's lab where we have 40 students who work on their experiments and on their research projects as well. So a very similar kind of lab as you saw just now for the undergraduate lab. So very similar. Once you come out, you will see that there is a passage. These are our incubators where microorganisms are grown at specific temperatures. So we have incubators of different temperatures where you can grow different microorganisms. You can also see that we have a very elaborate book bank which is managed, run by students totally, wherein we have lots of microbiology and other biology related and even computational books that you will see, which students have access to. You can refer to these books, and after a while Varsha ma'am will introduce you to the concept of book bank and the rules of the book bank as well. Obviously right now during the pandemic it has been very difficult to give access to the students to the book bank, but whenever you come to college you will have complete access to the same, and you can refer to the books that are there over here in the book bank. Now here you will see that there are two ways. One, you can go down, and another you can go up. Downstairs we have a specific enclosed lab wherein all our testing consultancy work related to pharma companies and food companies happens, and upstairs we have a research lab, a PhD lab. But before we go there, this is the area where the staff room is. Okay, so as you move in, you will see that straight you walk into the staff room where you find all the microbiology faculty working on their respective desk, and you will find your teachers of microbiology over here, and in case you want to communicate and talk to them, you can walk in at any point of time over here. And this is our instrumentation room wherein we have some instruments kept. So as you can see, there are instruments and computers kept over here wherein our students have permits to come and work on these systems whenever they want to do some search. This is an area where there is a laminar airflow. You can also see there is an ELISA plate reader. So when you do ELISA and you actually want to get the readings and quantify, you have a Biorad ELISA plate reader. We also have instruments like UV spectrophotometer, wherein you can do some chemical assays when you need to. And then there is a laminar airflow. This laminar airflow is a cabinet where you work with microorganisms, especially if you're working with microorganisms where there is some risk involved, and they are considered to be a biohazard. Then in those circumstances you work in this biosafety cabinet. And also, if you want to avoid contamination because it's a very sensitive strain, you can anytime work in this laminar airflow. We have at least 5 of these laminar airflows in the department. And the adjacent lab that you can see in the instrumentation zone is the Microfluidics lab, wherein we do lab on paper and lab on chip work. So this is another hood, another airflow that you see. And this is the lab on paper, lab on chip section, wherein there are a lot of research students who are working on developing faster diagnostic tools to detect microorganisms at a faster rate on a paper or on a chip, because this is the time when people want to come up with miniaturized diagnostic techniques. We also have fluorescence microscope over here, which is a very expensive instrument wherein you can see all cells fluorescently tagged on the screen. So this particular device that you see over here is a fluorescence microscope, which you can make use of when you come over here, when you want to see specialized cells. You can do that. So that's about the instrumentation room, where right now we are sorting some of the books that we have got from our alumni for the book bank. And now I will take you to the next lab, which is downstairs, which is our lab where we do all our analysis work, especially when we do some consultancy and there are industries which give us some projects. We do it in this zone, which is called as the clean room facility. So let's get in to the clean room facility. So this is in the ground floor, so you can see that there is a direct entry from the ground floor also. This opens up into the junior college quadrangle, and this is the room, which is a double doored room. So once you come in here, you have to sanitize yourself before you move in, because that area is totally sterile. It's kept totally clean, so you have to sterilize yourself with sanitizer, wear your apparel, and then you are going to move in. So as you move in, you can see another laminar airflow over here. We used to do some animal tissue culture work over here. There is an incubator especially for animal tissue culture if you are doing some animal tissue culture work. And this, you can see, opens up into the quadrangle. And this window is completely sealed because this is an enclosed region. Okay, and you can see that all things that are required to carry out any analysis over here is in the cupboard itself. Okay, so this is the area where we do all our analytical work. Now we will go upstairs and we will see the research labs. So here, these are the stairs for the research lab. So we are moving to the second floor now. So that's why I told you that we are spread across 3 floors, the ground floor. The majority of the labs are on the first floor, and then the research lab is on the second floor. So this is the research lab. We are getting into the research lab now. So this is where molecular biology research and PhD scholars work. So as you move towards the right, you will see that here the chemicals, the weighing balance, and you can also see a microwave oven kept. You'll be learning about the use of microwave oven for making some of the types of media to cultivate microorganisms in a microbiology lab. You can also see that there is an instrument called as a shaker, which is again used to cultivate microorganisms at a specific temperature, and by keeping it on a shaker, you can ensure that there is constant supply of oxygen evenly taking place through the media. There are also some specialized centrifuges, like a cold centrifuge wherein centrifugation happens at lower temperature. You can also see electrophoresis setup. I'm sure that you have heard of agarose gel electrophoresis in your 12th standard in DNA fingerprinting. So you can see here that there is a setup for the same. You can also see some other instruments kept over here which are required for experiments. This is a water bath, and this is a smaller version of the autoclave that you saw earlier. Here is Dr. Mayuri Rege, whom you saw in the orientation, and here is her PhD student, Ms. Mugdha Bilwalkar, who was also a faculty member at the department. Now we will go inside, where you can see that in this particular room we have A thermocycler. What's a thermocycler? A thermocycler is an instrument which is used for running polymerase chain reaction. Okay, and whatever gels you run for agarose gel electrophoresis, you can visualize it using this big instrument called gel doc, and you can take amazing photographs of those gels, which you can use whenever you wish to publish your work as research paper. This is a minus 10 degrees Celsius refrigerator where most of the microbial strains and some significant molecular biology based enzymes are stored at lower temperatures. So this is for that, and you will see that as you move out there is another room. So this room is basically where again we have a laminar airflow, as you can see. So this is another laminar airflow wherein you can see that there are apertures through which you can put your hand and you can work inside. So this ensures that you are not exposing yourself to any microorganisms that you are working with in this particular zone. So whatever instruments, other instruments you need while you are carrying out the work over here, that can be placed here itself so that you can very easily work in this zone. So this is the research lab. As you move out, you will also see that we have a machine which generates ice, because a lot of molecular biology experiments happen at colder temperatures. So this machine generates ice which can be used while you are performing the experiment. So this is the overview of how the entire department looks like.